This covers Unit 2, Key Area 5 of National 5 Biology, Transport Systems in Plants. Plant organs are roots, stems and leaves. We're going to start off by looking at cross-section of a leaf. Cross-section of a leaf is when you cut through a leaf thin ways and have a look at what parts you would find in there. At the top you've got the waxy cuticle, which prevents the plant from drying out. Below that you've got the upper epidermis, which is just a layer of ordinary cells that are see-through, let light through, to the palisade mesophyll. The palisade mesophyll gets its name, palisade, from the cells standing up on their ends, and mesophyll is a word to do with plant cells that do photosynthesis. So the palisade mesophyll is actually the main area of photosynthesis in a plant. It's got lots of chloroplasts inside each cell, and those little chloroplasts move round so that each one gets a turn at being near the sun. Below the palisade mesophyll, you've got spongy mesophyll. So the name mesophyll telling you there's photosynthesis happening there. You can see there's chloroplasts inside them, inside the cells. There's also lots of air spaces, just like in a sponge. Down at the bottom, you've got the lower epidermis, which is very much like the upper epidermis, see-through cells, but you'll also find guard cells there. Guard cells have got chloroplasts in them, so that means they can do photosynthesis, but that's not their main function. Their main function is to open and close pores, which are called stomata, and that pore lets gases in and out of the plant for gas exchange, including water vapour. There's also veins in the leaf. Veins contain xylem and phloem, which we'll see a little bit about later. Here's a table that runs through the different parts of the plant leaf. If you want to revise for this section properly, you want to be able to draw these cross-section of a leaf from memory. So draw one out and then learn the parts, and then make sure you can draw it without looking, and then you know that you know it well enough. The waxy cuticle prevents the plant from drying out, stops water evaporating out of the epidermis, the upper epidermis. The lower and upper epidermis have transparent cells, and they are protecting the more delicate cells inside, so the mesophyll cells and so on. Palisade mesophyll is where most photosynthesis happens, and it's got the most chloroplasts. Spongy mesophyll also does some photosynthesis and has big air spaces in it to allow the gases to move around. And these stomata, which are the pores, are controlled by guard cells, which open and close the stomata. Plants make sugar in photosynthesis, and photosynthesis happens in the leaves, and that sugar has to travel all around the plant by going up and down the stem. Sugar is transported in cells called phloem, which are living. And phloem cells are a tissue that have different types of cell within it. They've got these ones here, which are called sieve tubes. The sieve has small holes in it. This is called the sieve plate, and the cytoplasm can move between one cell and another through these holes, and plants transport food through the phloem. The other part of the phloem is these companion cells. Companion cells are where you'll find the nucleus that controls the whole thing. So together your sieve tubes and your companion cells make up the phloem for carrying food. Transport of water is a very important concept in this particular part of the course. When a plant takes in water through its roots, it's not just water that's coming in, there's also minerals that allow the plant to produce other things like grow its shoots and its roots, make fruits and flowers. Minerals are involved in that. So water and minerals are coming up into the plant, starting off at the roots. The water will then go up the stem and finally to the leaves, where it will evaporate out the stomata. Let's look in a bit more detail. So if we're dealing with our roots, there are some specialist cells in the roots called root hair cells, which have large extensions to give a big surface area. Water will go into the cell by osmosis through the root hair. There's always going to be a higher concentration of water outside the cell than inside. because water is going to travel up the plant and evaporate out through the stomata. So water is entering the root hair cells by osmosis. When the water goes up the stem, it's going up through xylem vessels. Xylem are dead cells which have lost their end walls and they're like empty hollow tubes, so they've got no cytoplasm inside. And xylem are for water transport up the plant. Water's always going up the plant. Xylem vessels also contain lignin round them to provide support for the stem and to help it withstand the pressure of the water going up. So sometimes xylem vessels are described as being lignified. The lignin provides them with some strength. So xylem are empty hollow tubes made from dead cells and water goes up them. When we get to the leaves, we're meeting a new term, transpiration. Transpiration is the water evaporating out through the leaves and it's coming out through the stomata. There's a couple of photographs over here showing guard cells and stomata. Here is the stomata, or stoma singular here. This stoma is open because the guard cells are fully turgid. When guard cells have plenty of water in them, that forces the stomata to be open. And when they lose water and become plasmolized or flaccid, the stomata close, and that prevents water loss from a plant that's already lost water. You don't actually need to know this mechanism of how they open and close. That's not required for National 5, but you do need to know that the guard cells control the opening and closing of the stomata. So transpiration is evaporation of water out of the leaves, and the water comes out through the stomata and it's all controlled by guard cells. When the guard cells are flaccid, the stomata are closed, and that happens at night. You're not going to need as much gas exchange happening at night because you're not doing photosynthesis then. If it's a very hot day, the plant might be in danger of losing too much water and it might close its stomata. However, if you've closed the stomata, then you can't really get much carbon dioxide coming in, so photosynthesis will be reduced. Now, one of the areas of the National 5 course that came down from higher is transpiration and factors that can affect it. So here they've picked out things that can increase transpiration. Rather than memorising them off by heart, it's much better to have a sit down and think about what transpiration is and what would cause it to increase or decrease, and figure it out for yourself at the time. Because in the exam they're going to want explanations as well, they're not just going to want lists. So let's do some imagining of leaves with stomata in them. What we're looking at is, what is the difference between inside the plant and outside the plant? If you've got a high concentration of water inside and low concentration of water outside, water is going to diffuse out through the pores. And it is diffusion because there's no membrane there, it's just a gap. So water will diffuse out if there's a lot of wind. Let's think about that. If some water is diffused out of the plant and it's windy, all that water that's diffused out is just going to get blown away and be replaced by dry air. So you're going to always have high concentration of water inside, low concentration of water outside. So a windy day will increase your transpiration. If you've got a lot of, or a high light intensity, the plant's going to be wanting to do photosynthesis, and the stomata need to be open in order for you to have photosynthesis happening, because you need to get carbon dioxide in. If it's a high temperature, the plant is going to be losing water to cool itself. If you've got low humidity, what does that mean? Well, humidity is water vapour in the air. So if there was hardly any water vapour in the air on a low humid day, then that means there's going to be a higher concentration of water inside than outside, and the plant's going to lose water on a low humid day. So those are ones that you need to take a bit of time to think about for yourself. The last bit of this section is introducing the bubble potometer, which is a lab experiment that you can do to investigate transpiration from a leafy plant. And there are different types of potometer, but the general principles are the same. What they're looking at here is there's a little bubble, and if there's a lot of evaporation of water from the plant, then water is going to be sucked up here and the bubble will move along. So there's different types of apparatus. Some of them you would add water back in to put the bubble back to where it started. Other ones you might just measure the distance the bubble's moved. These experiments often involve you having a control experiment as well. And a control is a copy of the experiment that's identical in every way, apart from the most important one. And for a control for this one, you might have a plastic plant. And why you would be doing that is to take away any effects of just normal evaporation, because if you've got water sitting in a container, it could evaporate away. So if you've got your control set up with a plastic plant in it, you can compare how much water did that one lose compared to the actual experiment, and you can subtract away any evaporation water loss. So make sure you know the name potometer, that it's to do with transpiration, and have a look at the diagram